Okay, hello everyone and welcome to the first in our new webinar series, today looking at the role of machine learning in proteomics. Today we're joined again by Dr. Christian Munch, who is our guest chair for this webinar. I just have a few of the usual housekeeping points to go over before handing over to Christian, who will introduce today's speaker and chair the question and answer session at the end. So as always, we're using our Slack channel for questions and discussion, so please join us there to ask questions, share any thoughts and discuss the work. Slack helps us to prioritise all the questions that others want to hear the answers to, so please use the thumbs up to let us know those questions you'd like to hear answered. It also allows the speakers to then answer any other questions or follow up after the talks have finished. For those needing an attendance certificate for this webinar, there'll be details on how to get this after the last slide. And once again, we'd like to say a big thank you to the European Proteomics Association, the British Society for Proteome Research, and they've especially asked us to remind any students attending today's talk that the Society is free to join for students and for everyone. The membership benefits include travel bursaries and conference discounts. We thank the Young Proteomics Investigations Club and the London Proteomics Discussion Group Committee for their help in support in setting up this webinar. And thanks also to the London Biological Mass Spectrometry Discussion Group, the London Metabolomics Network, and the News in Proteomics Research blog for promoting this event. And we're very grateful for Imperial College London for providing us with webinar support. Huge thank yous, of course, to our speaker and the guest chairs for their time today. And we're pleased to announce that our next webinar will be on Friday, the 24th of July at 2 o'clock British Summer Time, where we will revisit the COVID-related talks and we'll be hearing from two new speakers, Professor Andreas Pickelmeyer and Dr. Jeroen Demmers on this work in this area. So now I'm going to hand over to Christian, who will introduce today's speaker. Over to you, Christian.
Yeah, hello. Thank you for having me here. It's really great to be here, and it's even better to be here to introduce Lennart Martens, because I think it's really a fantastic opportunity for all of you to listen to what he's been doing over the many years. So Lennart is a professor at Ghent University and the Flanders Institute of Biotechnology, and he's also continuing to work at the EMBL EBI. So I think this is really those two stations, his scientific career to large part. He started from very early on. It's unusual for bioinformaticians to work on proteomics from the early days on, and he has since his master's degree. And I think he has really done a lot for the field. So his career has been throughout split between Ghent and the EMBL EBI. It's been back and forth doing different things, and one of the really big contributions he's done to the field is starting PRIDE, the proteomics database, to make data available to the community. So his focus is on novel machine learning algorithms and using them for the analysis of mass spectrometry data and the application for large-scale processing of publicly available proteomics data. So with what I've told you, he's also accrued quite a number of accolades over the years, great things he's done, and he's, for example, a fellow of the Royal Society for Chemistry. And he's been very involved with HUPO and getting things organized there. And if you haven't seen yet, it's worthwhile checking out. He's also written 2 books on computational mass spectrometry that have been really useful and I think are a great resource for everyone to read and to understand better how we can use machine learning for proteomics. So with that, thank you very much, Lennart, for being here today, and I'm really looking forward to your talk.
Thank you very much, Christian, for this very nice introduction. I'm very glad to have all of you here today, and we'll talk a little bit about machine learning. And as Christian mentioned, it's a dual interest of mine to investigate how machine learning can help proteomics informatics, but then also how we can deploy this machine learning based knowledge and tools to reprocess public data, such as the data that is in the PRIDE repository. And so both things will come into play during my talk. So maybe one of the key things to take home from the current state of proteomics is a picture that you see here, which obviously is a submarine of sorts. Now you can see the periscope cutting through the waves there, but the big problem here is that while we can infer the presence of the submarine under the waves, we don't actually see the submarine. So we don't really know what kind of submarine it is, what it looks like, but we know it's there. And in many ways, this is how we how we look at the proteome today and have been looking at the proteome for the past 2 decades. The idea being that we see some parts of the proteome, but we are also keenly aware that certain parts of the proteome remain hidden from us. And so one of the nice things that we can do once we have machine learning tools available is that we can get a broader picture of all of this proteome information that is currently remaining hidden. And so that is what I'm going to be focusing on in my talk, how we are going to get a better view of the proteome through all these tools. So in overview, we're going to talk about the first big issue, which is that we are addicted to sequence databases in proteomics, which is quite useful in many ways, but is also limiting our view. So that is our basic problem we have to fight. Second thing is then that with machine learning, we can predict analyte behavior, and that in turn allows us to open up new information. So I'll be talking about ways in which we have been predicting analyte behavior using machine learning. And then I'm going to talk about a search engine that we have built called IronBot, which leverages all of these predictors to interrogate proteomics data in a completely new way. This actually gives us a whole new view on the proteome, including PTMs, and we need a mechanism to make that available to the world at large. And so that is the penultimate title there, is a dissemination of all of this new knowledge. And then finally, we also use some machine learning at a very high level to understand tissues at the proteoform level. So let's start with this addiction to sequence databases. So fundamentally, proteomics would be a wonderful place, and it wouldn't need much bioinformatics if every spectrum looked like this one. So if the sequence is peptide, as you can see here, we have a full series of B ions that completely cover the sequence, and we can literally ladder sequence the peptide by jumping from the 0 position to the first peak, which gives us the proline, and then on to the glutamate, another proline, threonine, and then onwards to isoleucine, etc. Now, of course, we know that this is not what a spectrum looks like, because at the very least, we have to include all the Y ions as well. So they're given here in teal. Now, even then, with this confusing combination of two different ion series, still this would be very easy to sequence. But of course, this is not what real spectra look like. Real spectra look more like this spectrum, which means that a lot of the peaks that we wanted to see, the B and Y ions, they're actually gone. They're not recorded for whatever reason. There's a few red peaks there that are interfering peaks that have nothing to do with the sequence of the peptide. We don't really know where they come from, but they're there and they're annoying because they confuse us. And even if we were to jump correctly to all the consecutive B ions that are left, you will see that in the first jump, we end up on this blue peak that is labeled PE. So the real sequence is PE, proline and glutamate, but the problem is they could be reversed as well, because that would give us exactly the same mass. We would then have the glutamate followed by the proline. And then, and I'm not entirely sure this is accurate, but imagine that two isoleucines would have the same mass as a glutamate proline pair, then you would have another problem, because now II is an option as well. And then, of course, all of you know that an isoleucine and a leucine are exactly the same chemical composition, which means that they have exactly the same mass. And so as a result, II could also be LL, could be IL, could be LI. And what you can see very clearly is that the limited information in the spectra causes ambiguity. We now have multiple options for each of these peaks, multiple equivalent explanations. And of course, depending on what your spectrum looks like, this problem can get better or worse. But fundamentally, we can't read a sequence straight from a spectrum the way our nucleic acid sequencing colleagues can, purely because we lack most of that information that we need to get a unique sequence derived from a spectrum. So the big question is, how do you fix this problem? Well, the field very quickly settled on the way that we will fix this problem, and I generally dub this cheating. It's cheating in a very clever way. But essentially, we resolve the whole ambiguity by saying of all possible peptides that could be encoded by this spectrum, we're only going to allow those peptides that we expect to find in the sample. So if you have a human sample, then the peptides that you expect to find in the sample are all derived from the human genome. And that is where the dependence of proteomics on genomics comes from, because we really do need to have all the genomes in order to be able to get this list of potential peptides that generated our spectra in the instrument. And so what you can see here is a flow chart of how this works. From the database, you do an in silico tryptic digest, which yields a bunch of different peptide sequences. Each of these peptide sequences can be then transformed very easily into a theoretical mass spectrum with all the B and Y ions that we can possibly expect. And then these are matched to the real spectrum, which you can see at the bottom, the experimental spectrum. And then the final result is you get peptide to spectrum matches with a given score. We rank them by score, and then we pick the top ranked one, and then we evaluate that downstream for false discovery rate corrections. But fundamentally, this is how we work. We need this catalog of spectra in order to match our experimental spectra against, so that we can then derive scores. And even if we use spectral libraries, that effectively boils down to the same thing. We have a library, a limiting set of spectra here, usually less spectra than there are theoretical peptides, because it's a smaller number of peptides that we have seen so far in the mass spec, and then we use that kind of matching, so it's the same principle. Now that's all well and good, and it serves the field really well for the past 20, 25 years or so. But there is a very important assumption here, and the assumption is in the theoretical spectra. And as you can see, all of these peaks are equivalent in height. This essentially translates into the assumption that every fragment ion is equally likely to occur in a spectrum, which we know for a fact is not the case. So we simplify the situation, but it's not really a big problem, because this list of potential peptides is actually reasonably small. It's on the order of 1 or 2 million for a normal eukaryotic organism, which is actually very small compared to the total space of all possible peptide sequences that you could make that would potentially be there. Now, this is all well and good when you do a proteome, when you investigate a proteome of a single organism. However, as soon as you try to do something fancy, which is what we all try to do these days, like proteogenomics, metaproteomics, or open modification searches, this problem starts to become really tangible again. Because instead of a very limiting filter that we set on our potential matching peptides, we now have a far larger space in which we have to search. It creates computational problems in the sense that you have to search many more sequences, so you have to speed this up somehow, but it creates a lot of scoring issues as well, discrimination issues. So how do you figure out whether peptide A or peptide B is the actual match? So I'll try and illustrate that based on the same workflow. So imagine you now have a very large database because you're trying to do a 6 reading frame translation of the human genome, or because you're doing metaproteomics, or because you've added open modifications. So you've allowed thousands of modifications on every single residue, essentially. What happens now is, instead of a reasonably manageable, quite well filtered list of potential peptides, you now suddenly have a whole bunch of peptides. So you're thinking about 2-3 orders of magnitude more peptide candidates, maybe 5-6 orders of magnitude, depending on how crazy you get with your situation. That in turn translates into a far larger number of theoretical spectra. And now we get into trouble, because as soon as we start matching these experimental spectra to our theoretical spectra, we start seeing these kind of scores. So these are all different peptides. As you can see, they differ here in their final amino acids, and then I also replaced valine with I flipped around the alanine and valine here, and as you can see, all of these options are actually pretty good matches. So now our search engines start to fail, because our search engines essentially tell us that these different peptides are all good matches. They're all good candidates to match with this experimental spectrum. And what we have is we have the ambiguity we started out with. It's coming back, and it's kicking us in the behind. It's essentially telling us your spectrum does not have sufficient information to distinguish between these 3 candidate spectra. As a result, we lose a lot of identifications. And this is really problematic because, of course, everybody wants to get some proteogenomics information, wants to do metaproteomics, wants to do open modification searches, because we know there's a lot of information there that we're currently not seeing because of the limits set upon us by our database addiction. So the rest of the talk will be about how do we circumvent that, and what do we do with the results once we've managed to circumvent that. So the key way, of course, as you will have expected, that we try to tackle this is by predicting analyte behavior. So we're trying to change the way we look at the data, and we try to get rid of these shortcuts and these incorrect assumptions that we use, and replace these assumptions with pretty accurate predictions of how a peptide will behave, how it will fragment, how it will behave on a liquid chromatography system, and how we can use this information to more accurately pinpoint which peptide is the real one. So the first thing we did, and we started with this a long time ago, actually the first publication is from 2013, and the first actual version of this algorithm is from 2011, which is a very, very long time ago. And we called this MS2PIP, and it is a predictor for peptide fragmentations. So here is a typical example. This is the experimental spectrum as shown in the peptide shaker tool. And then this view here is the annotated spectrum. So you can see it fragments really nicely. It has lots of Y ions, quite a few B ions. Now, when we ask MS2PIP to predict the spectrum for the exact same peptide, this is the prediction that you get. What is interesting is that the prediction is pretty accurate. If you just look at the tail here, for instance, or rather it's the head because it's the high Y ions. You can see the Y10 is the prominent peak, and then you see 3 peaks, 2 of which are kind of in the same ballpark, Y11, Y12. Y12 is a little bit taller than Y11, and then Y13 is the lower of the 3. It's reflected extremely well in the prediction. So we've been upgrading and extending this algorithm, and I'll show you some more recent results. And of course, as you know, other people have now started doing this as well, right? So in 2018 and 19, new algorithms came out that used even more sophisticated methods of predicting certain aspects that tend to do slightly better than MS2PIP. But interestingly, if you read the papers and you look at the applications, you will see that there is a law of diminishing returns at work where all of these algorithms now appear to be sufficiently good that it doesn't really matter downstream which particular one you use. So that's really good because now you actually have a nice choice of spectral prediction algorithms. And we like to think that we were in the vanguard, one of the first people to seriously start exploiting this information. Although, and in all honesty, this must be said, other people did it before us as well. We just like to think we're the first of the modern people, which is one way to slice this, I guess. Anyway, we can actually also investigate whether or not our models are sufficiently transferable. And so what you can do is you can see whether you can train models for one approach, say CID fragmentation, and then whether you can transplant these onto HCD models. Now you can see 2 models here. This is the CID model performance, the HCD model performance, and these 2 models perform roughly equivalently. However, if you look here, this is where we try to apply each model to each type of data. So the blue line is the CID model. So the CID model predicts the CID data pretty well. And we already knew that. That's what you see on this box plot here on the left. However, when you take the HCD model, which is in orange and which predicts the HCD data essentially equivalently well as the CID model predicts the CID data, the HCD model does a pretty poor job of predicting the CID data. Now that makes sense because CID is a very different way of fragmenting. It's a much lower energy way of fragmenting. And so as a result, the specific model performs much better on the specific data, whereas if you start crossing models with data, it actually gets worse. You see the similar trend here with the CID model doing a pretty poor job on predicting HCD spectra. Now, that is very simple. Everybody kind of expected this. However, it also works from modifications. So here, for instance, we look at TMT and iTrack. So if you take the HCD model, which is pretty good, and you apply it to TMT labeled data analyzed with HCD, you suddenly see a dramatic drop in performance as well, almost on par with the drop you would see going into CID. And the reason for this is that the TMT label dramatically affects the way that quite a few of the peptides fragment. And so as a result, we can build a specific model for TMT peptides in green here, and you can see that that green model performs really well. It actually outperforms the HCD model, probably because the TMT model actually can grasp onto very specific things that TMT induces, which are more conserved across sequence than the HCD fragmentation, which is more sequence dependent. Now, another interesting thing is that we can build an iTrack model as well. And so, of course, TMT and iTrack are chemically quite similar. And so as a result, you see that the models actually cross-perform with roughly the same performance. So whether you train on TMT or you train on iTrack is really not that important as long as you train on this particular type of modification. And then what you can see here is when you have a model that combines iTrack with phospho groups. So this is a phosphoproteomics experiment. So quite a few, although not all of them, quite a few peptides carry phosphorylations, and these phosphorylations actually do affect fragmentation as well, to the extent that most of the models are now a little bit worse. So the TMT and iTrack models are a little bit worse here, and a specific phospho model is slightly better. And it's kind of hiding the fact here that a lot of the phosphopeptides might actually not fragment that differently, but some of them definitely do, and as a result you see quite a big difference. So what is this telling us? It's telling us that you have to adapt your models to specific types of, say, modifications or different types of analysis. It's kind of obvious, but I guess we were the first ones to show it that clearly. Now, here's an example of how dramatic the difference can be. You see in blue at the bottom an empirical TMT spectrum. So this is the actual spectrum coming off the instrument. And here you can see the HCD model predicted by MS2PIP. And as you can see, while the peaks are roughly equivalent, there are substantial differences between the green and the blue spectrum. Yet if you look at the TMT model of MS2PIP, now you see it does an extremely good job of predicting exactly what that spectrum will look like. Interesting for those of you who do TMT or iTrack labeling is that very often you see a drop in identification rate when you use these labels. It's not enormous, it's between 5 and 15% depending on circumstances, but one indication of why this could be is TMT and iTrack seem to lower the number of recoverable fragment ions. So you see fewer blue ions, especially fewer prominent blue ions, then you see green ions, which are a good representation of what a typical HCD fragmentation of the same peptide would look like. Given that many of our search engines actually count spectra one way or the other, there is a fairly good chance actually that these blue peaks lead to a far less accurate identification or a more ambiguous identification than these green peaks, because here, of course, you have much better sequence coverage. Hence, it could very well be that that is precisely the reason why we find fewer TMT identifications than if we were to analyze the same sample without the TMT label, just using HCD fragmentation. Right, so we've got MS2PIP, we know a little bit about the background. Now the question is, where do we position it in our workflow? Well, it's actually quite easy. You position it right here at the level of the in silico fragmentation. So instead of using these theoretical spectra where all the peaks are equivalent and you just predict all of them and somehow you think that these very weird and unnatural spectra will match pretty well to your experimental spectrum, you now actually predict accurate spectra. And these accurate spectra now allow you to distinguish between peptides that originally seemed equivalent. And the effect is quite dramatic. So these non-distinguishable peptide scores suddenly become quite distinguishable, because if there is a big peak here and a big peak here, it must be that spectrum, sorry, that peptide, because for the other two peptides, the big peaks are actually elsewhere. And so the correlation now becomes a very important measurement. Now, of course, we didn't just speculate on this, we actually managed to prove this, and this was work that we did with Jonathan Blackburn of University of Cape Town in South Africa, who was working with Mycobacterium tuberculosis. As you know, South Africa, because of a very large amount of HIV positive and H patients, had a serious problem with human infections of all kinds of mycobacteria. And you can see here several of them, because immunocompromised people, of course, can even get bovis and avian infections. And he was trying to study these, but he had to cobble together a database of sequences that essentially consisted of RNA sequencing data across the whole mycobacterial family, trying to get as many possible predicted sequences as he could get, because again, we're addicted to sequence databases. So when he was searching this with normal mascot, which is kind of the reference, even though mascot is starting to show its age a little bit, you can see that you get a reasonable number of proteins identified, but he was not very pleased with that number. So then he tried something a little bit more fancy, which is Crux, and Crux is essentially sequest plus percolator, especially at that time. And then you can see that you can essentially double the number of identifications. So red versus blue is about double the IDs, which is really showing you the power of machine learning again, because Percolator, made by Lucas Schell, actually manages to leverage machine learning to triage the identification results into definitely good results and definitely bad results in a much more nuanced and a much more successful way, if you like. Now, still not entirely happy with these results, Jonathan, who knew about our work, then challenged us to try the same thing with our predictions of spectral intensities. And so we did that and we employed those. And now you can see the green curves coming up, and the green curves add another 50% or so, or maybe 30 to 50% on top of the percolator results. Now, that is very, very nice to see, and it shows that this additional information really helps. Now, one interesting point here is the Beijing strain. And the Beijing strain of tuberculosis, you can see that the percolator and the rescore, so using the MS2PIP information, are kind of matched. For all uses and purposes, they're equivalent in the number of hits. However, if you look at purple, which is the grand total across all the identifications, you can see that purple is much higher than you would expect. And the reason for this is that rescore results, our results, do not agree with the percolator results. So in fact, because we have the spectra predicted quite accurately, we assign a given experimental spectrum to another peptide than Crux does. Now, if you're confronted with this, of course, you have three options. One, Crux and rescore are both wrong. Second, Crux is wrong and rescore is right. Third, Crux is right and rescore is wrong. So what Jonathan did was, and I have to stress this, he did this behind our backs. He went to Rudi Ebersole in Switzerland and asked them to do MRM runs to validate the presence of about 20 peptides in the samples, and these were all uniquely identified by our approach, by rescore. So at one point I got a phone call from Jonathan. It was in the morning in Belgium. I pick up my phone. He says, "Hi Lennart, it's Jonathan. How are you?" I said, "I'm fine." He said, "By the way, we did these MRM tests because we didn't really believe the results coming out of your search engine." So I can tell you my blood pressure dropped quite dramatically because it's a very serious test of your algorithm that you don't expect. But it turns out every single one of these MRM assays was successful across all the relevant samples. So the point was that the rescore identifications were validated, despite the fact that they disagreed with the Crux identifications, again showing that this is a promising approach. So I think we were one of the first, if not the first, to actually show that this disambiguation power of predicted spectra is a reality in real life. Now, we actually took this a step further. So we took rescore and we built it into a fully machine learning driven search algorithm. And the way we do this is we essentially use percolator. So we didn't reinvent the scoring engine for this particular approach. We just used the existing percolator by Lucas, and then we compare it to MSGF+, and we can use normal percolator, and you can see that if you combine MSGF+ and percolator on this particular data set or on that particular data set, you can see that actually percolator adds a few unique identifications, most of the identifications are shared. This is what we expect. Percolator makes a good search engine better. However, when we give percolator only MS2PIP features, that means no search engine features whatsoever. You actually get still more identifications out of it than you would have gotten from MSGF+ alone. And it's very comparable, albeit a little bit less than what you get when you use the search engine together with percolator. And then if you give the percolator both the MSGF+ features as well as the MS2PIP features, then you get the largest number of identifications. But for all uses and purposes, it is essentially almost equivalent, down to a few hundred identifications in, say, 25,000, whether or not you use a search engine at all. So by just taking MS2PIP results and percolator, giving it a bunch of peptide candidates, you can actually build your own search engine. It is a machine learning trained search engine, which again, I think we were the first ones to show this. And of course, it doesn't depend on the data set because all data sets give you essentially similar results. Now, what was interesting was that we then investigated the weights of the features that percolator hands out. And so essentially a weight means how important is this feature to distinguish good from bad hits. And if you use search engine related features, most notably here the e-value score that the search engine gives the PSM, what you can see is it becomes a pretty important feature, which makes sense because the search engine is already doing the triage for us to some extent, right? That's why we use search engines. And then the other parameter that was quite important is the peptide length. So percolator is building a score on the fly based on the search engine score, which makes sense, and the peptide length, plus a limited amount of other features as well. And so a lot of the features that you're giving to Percolator essentially are pointless because Percolator is weighing them at 0. So it's essentially not using these features in the calculations. However, when we remove the search engine information and we go only with MS2-PIP derived information, we see a completely different picture. Now, a lot of the features, including the new ones that we add, are being used by the scoring function. So the scoring function now uses a broader set of features, a broader set of characteristics to identify the spectrum than previously. Now, that is all well and good. Why is that important? It is important because this feature here, the search engine score, is predefined. So somebody comes up with a scoring function and says this scoring function shall now work for all possible data. However, if you have special data, a special instrument, or something else that is not exactly the same as what this scoring function was meant for, the scoring function does not adapt to your situation. Here, this thing, it can adapt to your situation because it is learning from your data on the fly, based on target and decoys, what your data looks like and which features are important for your kind of data. And so that is a very powerful thing. We are no longer limited to predefined and potentially heavily biased scoring functions for our data. We can in fact make use of a learning system that adapts to what your data looks like, and it does so in a controlled fashion because the results you get back, and that's what we showed here, are very, very similar to what you would get back with a normal search engine and are extremely competitive even to a normal search engine plus Percolator. So this is reliable data, this is very good data, and it is highly adaptive, much more adaptive than anything we've ever built before. So that's a quite crucial thing to keep in mind. Now, with all of this, we've just only looked at prediction of MS2 intensities. We can also predict other things. So one of these other things that we have been predicting is retention times. And of course, retention times have been done to death. What hasn't been done, however, is retention times for unseen modifications, because we want to find PTMs. And if you want to find PTMs, you do not know in advance necessarily what the retention time effect is going to be of every single modification in Unimod, of which there are 1,500. So we built a system that no longer is trained for a specific modification, but it tries to learn generically how does chemistry affect retention times. I'll not go into all of the details here. You can read all about it in this bioRxiv manuscript, which has seen quite a lot of attention, so maybe you've already read it. The bottom line is that we start looking at the chemical composition of everything. So rather than amino acids and modifications, we just look at chemical compositions, and then we train a rather complex deep learning network to assess what the effect of a particular chemical composition is going to be on a retention time. Some results. If you do normal retention time prediction, so without taking into account any special modifications, you can see that we've built a pretty good retention time predictor. This is observed versus predicted retention time for 3 different data sets with different kinds of gradients. And what you can see here is the median error, and that's about 0.33 minutes, and the delta T 95%. So this is where 95% of the deviation is, and it's within 0.9 minutes for our algorithm with an R squared of 0.997. If you look at what is probably the steepest competition right now, DeepRT+, which is also a deep learning-based retention time predictor that was recently published, you can see that they fare slightly worse on this data set, but the difference is negligible almost. This is a data set for which we don't have a competitor's assessment, but it's also pretty good. You can see the errors are on the order of about 1 to 4%, depending on whether you want to take the median error or the delta 95. So very, very good prediction. And then here again, a comparison with DeepRT, where the results are essentially similar. So the R is a little bit better here for DeepRT. The delta T95 is a little bit worse. But again, on the whole, this thing is as good as the state of the art when it comes to normal peptides, which is what you would want, right? However, what DeepLC can do, and what nothing else can do, is it can predict the effect of a given modification. Here you can see the retention time prediction for dioxidized peptides. So peptides where the methionine, for instance, has been doubly oxidized. And typically, if you don't tell the prediction algorithm about the double oxidation, it's going to be off, because the double oxidation actually gives it a slightly larger retention time predicted than it has in real life. And so it's above the line where you would want it to be. Note that as the retention times get longer, that difference becomes smaller. That's probably because it becomes less important, because the peptide is so hydrophobic, it doesn't really matter whether it's oxidized or not. And also this is more or less where the wash starts to come off. So you can see this effect here as well. However, when you tell DeepLC, look, this thing is dioxidized, and maybe it's never seen a dioxidized peptide before, it has seen an oxidized peptide maybe, but never a dioxidized peptide, suddenly it actually recenters the predictions, and it actually gets these things right. So it figures out from the chemistry that this is the way the peptide should elute, and it turns out it corrects it the right way. You can also look at the opposite effect, for instance here, formulation. Formulation actually means we predict it at a time that is a little bit too early, because the formulation makes it slightly more hydrophobic in real life. And so again, if we don't tell the algorithm the fact that the modification is present, it's going to make the mistake that all algorithms today would make, and it's giving it a slightly too early retention time. But as soon as we tell it, there is this thing called formulation. You have never seen it before, but there is this thing and it looks like this chemically. Suddenly it can predict very, very accurate retention times. One thing that you may also notice is that the spread on the data points is reduced when we give it the right modification. So not only is it giving us a better position, so it's giving us the correct retention time, the error on that prediction shrinks at the same time. And so you get an R that is quite substantially better, and you get a median absolute error that is quite substantially better. And so this is really a breakthrough. Nobody's ever done that before, but it's really, really powerful. And I'll show you later on how powerful this can be when used correctly. You can also do another test. I'm not going to waste too much time on this, but it's an interesting test because we don't have that many gold standard modifications that we can test against. We can actually tell the algorithm that, say, a methionine is in fact a glycine that then carries some extra atoms, right? So we can never show it a methionine at all to train on, and then tell it that a methionine is a glycine plus modifications because it has seen glycines before, but it has never seen these modifications that make it into a methionine. And then we can see how well it predicts the actual retention time. And what you can see is anything that is above the line here is actually better predicted when you encode the amino acid as glycine than if you were to encode the amino acid normally. And then everything that is below the line here, which is a zoom, by the way, the right-hand side is a zoom in of this colored area, you can see these things are actually predicted slightly worse. What you can see is that the large effect is it is as good or better, and then better is actually quite significantly better than normal. And some are worse, but when they are worse, the differences are minor. So the thing actually works really, really well. Now, DeepLC is very interesting, but it's not going to solve all your problems because everybody has slightly different LCs. Now, the problem in proteomics is slightly less dramatic than the problem is in metabolomics, because in metabolomics, of course, the metabolites have very different chemistries and therefore require very different separation techniques. And so here, for instance, we compare the retention times of two different data sets in metabolomics, and you can see this is the fit to align them. It's very different. Now, there's not a linear fit, it's a complex nonlinear fit. And this is a really big problem, especially in the world of small molecules, but it also applies to proteomics, where if you try dramatically different ways of separating things, you're going to get very different retention times. And as of now, nobody has been able to predict an accurate retention time if you have this input data and you have to predict for that kind of output. And so that has been very difficult. And the result is that retention time prediction has not been used very dramatically because it's difficult to adapt somebody's predictions to your own workflow. In proteomics, it works better because we tend to all use C18 and the same kind of gradients. But in metabolomics, this has been a serious issue. So what we have done is we've built system called CalLC, for calculate all LCs, where we can train based on different data sets on different setups with multiple machine learning models for each of these sets to set up combinations. And then when we want to predict for a new setup, we can calibrate each of these models to the new setup. And then we can see how well does the new setup correspond to the prediction. So how does the calibration work? And so for set X here, we can see that the model calibration on, say, set 1 is actually pretty good. It seems rather linear. For set 2, the calibration is much more complex and the errors are much worse. So set 2 and setup specific model 2 are a poorer fit to our unknown data set than set 1. And then we make a weighted average of all these models, where set 1 has a much larger contribution to the final model than set 2, because set 1 is a much better fit. And that gives us the power to predict retention times across setups. But still, and this is very important, despite the fact that set 2 is a reasonably poor fit, it still helps to improve the model that we get finally, because there is a certain fit and because it can attenuate some of these predictions and make them better. I'll show you that on the next slide where we evaluate this. So if you look at a setup specific model, it actually performs really well. This is the average error, the lower the better. And so it performs reasonably well. If you take the best calibrating model, you get a much better result. And then if you take all data that you have and use that to make the prediction, despite the fact that some of this looks very different from the data set that you're trying to predict, you get even better results. And what is happening is that the system is learning from very unrelated systems how your analytes behave, even on this weird new system. And so the net effect of this is a much better predictive power, even if the number of training analytes is very small, because we're training now across many different analytes, across many different setups, and we're borrowing that knowledge and exploiting it here, which again is the first time anybody's ever done that, but it's clearly doable. The effect of this becomes less dramatic, the difference between these three different predictions, as we get more training analytes, which makes sense. If you have many training analytes, you can calibrate or adapt any system to work on any other system. But still, even then, because this model, which uses all the data, still has more input, it will always be better than the other ones. And so by doing this in metabolomics, you can actually start predicting, say, from a cation exchange chromatography, you can start predicting how a C18 retention time will behave for a given analyte. And we can show this again by comparing to a model that was from the Kohlbacher group, the Eichler model, and that was published in, if I recall correctly, 2016. And it's a very good model for lipids, but we can actually do better. And we tend to do a lot better when there are fewer training analytes. Why is that? Again, because we use all the data, including lipids that have been analyzed on completely different chromatography setups, but still they contribute and still they make this better. And then you can see a very similar curve where as you get more training data, the thing becomes better. Our thing becomes better too. You also see that the variation gets squashed, so it gets more precise in its predictions, and it always outperforms the old model because it has a lot more data to work with. So this is actually extremely interesting. It would mean that you can calibrate LC systems with a bare minimal number of calibrants. And that would mean that retention time predictions become way more portable than they are today. Plus, it means that you can build a better model by even when you use data from other people using different models than you do. And so this is, I think, one of these other things that show you that when you have lots of data and you have the right mindset to deal with that, your data richness translates into a capacity richness, into models that are so much better than if you were to ignore all of that heterogeneous data. Now, we've actually used, we've used all of this information in something that Maarten Danes in a previous webinar here for LPDG already talked about, and so I'm not going to go too deep into this, but the bottom line is that DIA, where you actually are reliant on spectral libraries most of the time to identify things, you can now actually use these accurate predictions, do a narrow window DIA to calibrate this, and then analyze wide DIA data and get better results. So these are the results that you can see in F here at the bottom, where we actually outperform any other system. So E is a system that, as you can see here on this plot, that is just going from the database and then calibrating based on that, which is a really bad system, so it's got very poor results. But we also outperform D, which is the most comprehensive human DDA-derived library, spectral library, and we actually outperform that because we can take the whole database, not just what has been seen before, but the whole database, predict the fragmentation spectra with MS2PIP, predict the retention time with a retention time prediction algorithm like DeepLC, then calibrate with narrow window DIA, and then apply it to the wide DIA data and get many more results. So this kind of stuff has again been proven in real life and does work really well. And again, it shows us that we don't have to be limited in our approaches, that these machine learning algorithms set us free from all these built-in biases that we might be dragging along from the past. Okay, so what can you do now, apart from changing DIA, apart from getting a few more identifications out of, say, a mycobacterium analysis? What can we really do with something like this? And so we've been building on this for several years now. We've built a new search engine called IronBot, which is completely machine learning driven. There is no more predefined scoring functions. All of that is gone. And when you use IronBot, you can search all 1,490 known modifications in Unimod and all possible single amino acid polymorphisms, and then analyze those all at once and see which of these are found in your data. And you can actually try it out online on this URL if you want to. It's kind of limited in the scope. It's only human data that is searchable, and it doesn't have all the enzymes, but it's just a tryout version that you can play with. And what we have done in the first iteration was we took the well-known human proteome from Pandey et al., which consists of quite a bit of data. And then we've analyzed that through IronBot, which actually is very fast. So we made it extremely fast. It can run 1.5 hours for 1 million spectra on our really poor data sets, on our really poor servers. And we found 368 of the modifications represented on 260,000 residues, 140,000 peptides, and 15,000 proteins. So we found lots of data in this well-known big data set. So if you look at the frequency of residues that appear modified, it all makes chemical sense. The N-terminus, which is a free amine, is extremely often modified by very many different modifications. Cysteines, which are highly reactive, are very frequently modified with a host of different modifications. Methionines are highly modified. Again, they're quite reactive, and so forth. You see all the chemically reactive residues here. They have lots of different reactions happening to them. Note as well that the lysine amine group is also highly reactive, of course. And then you can see here the less reactive with very few modifications, which are obvious because these are, of course, the small aliphatic ones. Now, when you see something like this, next question is probably which modifications do you find? Well, these are the top, I think, 30 odd modifications that we find. And you can see that in this particular data set, there's a huge amount of carbaminomethyl going on, and it's not just affecting the cysteine, so it's also affecting the lysines and other residues. Then you see oxidation, for instance, you see acetylation, you see methylation, you see, of course, also artifacts like ammonia loss or pyroglutamate. You see dioxidation, which I talked about earlier. You also see things like 4-hydroxynonenal, which is very interesting because that is, of course, a lipid residue that is oxidized. That is a clear indicator of oxidative stress. So you actually see, again, things you expect to find, and you can see here how often you find them and at what PSM rate you can find them. So a next question, of course, is then how accurate is this, right? So you can find all of this, but finding it is easy. Making sure it's actually there is hard. So we then try to analyze a data set that was published by Zolk et al. From the Kuster lab in 2018 in MCP, where they synthesized several peptides bearing modifications. And they here kind of repeated in a way this analysis that I showed as well, which is that you can predict or look at the real spectra, and you can see that some modifications actually mess with the spectra. And then here, the cool thing is we actually know what kind of modifications to expect, or at least that's what you would think. Because if you then take IronBot and you look at, for instance, all the biotinylated peptides on lysine, we see that we actually do find biotinylation as one of the most common modifications by far, but we also find other modifications like formulation on the serines and formulations on the threonines, which are probably artifacts. And then here you see an example of that, where you have a beautiful Y-ion series that very nicely shows us that this serine is in fact formulated with a very high correlation coefficient with the predicted spectrum. So you actually start finding a lot of things that you don't expect. And so again, we don't have a gold standard here. It's very difficult to say whether or not our predictions are right, but at least they look to be very, very accurate. So even synthetic data can be altered in a way. Now, we can evaluate the performance of IronBot when we compare with the results of MSFrag. And then you see here that IronBot in a normal search has more identifications than MSFrag because it has a lot of added sensitivity. And when you do an open modification search, they're almost equivalent. And then what we can do, by the way, this orange here is where IronBot says we actually have essentially equivalent matches. So IronBot is highlighting the ambiguity issue. But the ambiguity issue that we see here highlighted in IronBot is also present in MSFrag or any other search engine. It's just that because we are so sensitive to this, we actually highlight it when it's happens. And so what you can see is we get roughly equivalent results. And then here at the bottom, at the PSM level, IronBot always finds more in the normal search, slightly less in the open search. But when it comes to unique peptide sequences, IronBot tends to do better on the number of total peptides in both cases. So we find more peptides, more distinct peptides than MSFragger. And interestingly here, when you look at these MSFragger unique hits, IronBot actually identifies the same spectra. But the problem is that the same spectra identified by IronBot get a higher correlation and a higher or smaller, in this particular case, retention time difference than MSFragger, highlighting again that they could be better identifications than the original ones proposed by MSFragger. Now, which are the features that IronBot seems to rely on heavily? It relies on peak counting, which is what most search engines do. It relies on the correlation between the MS2 and the predicted and real spectrum, but it also relies quite heavily on this retention time error. And that is very important because we now have DeepLC, which is capable of testing whether a modification results in a retention time difference. And so it's clear from this plot alone that this retention time difference really helps to disambiguate a peptide carrying a modification from, say, another peptide carrying another modification, or the same peptide not carrying a modification. And then peptide length is important, the total number of peaks that are in the spectrum, the charge, and then the explained ion intensity. And so the score function is shown to be built like dynamically through machine learning. Now, if we look at the top modified proteins, these are glycolysis proteins. And that's interesting because that's their quantification issues with proteins. If you look at it, the number of modifications on these various proteins are quite staggering. And you see a list here of jar, and many of these are probably artifacts. Now, if you want to quantify one of these proteins, and if you then assume these modifications are actually going to be artifacts that are changed in ways that have nothing to do with biology, then this is a really big problem, because you're going to have differences between different analyses which are purely derived based on artifactual circumstances. So your technical variance is going to make a big difference in your quantification. And so if you find glycolysis proteins as deregulated, as significantly altered in expression level in your experiment, that may be a bad sign, because it could be that you're looking at an artifact. Another way to look at this is to take actin and see what the difference is between the PTMs we can expect in actin, because it has been studied for more than 60 years now, very thoroughly, and what we can actually find when we just look at the Panday data set I talked about earlier. So these are all the known modifications of actin, and you can see 2 lines in red. These are things we're not looking for, because cross-linking we can't find, because that's, of course, it's not really a modification, it's a much more complex issue. And arginylation was not in our list of modifications at the time. So we can't. And now you see a picture of actin with all the modifications highlighted. And now the question is, which of these modifications can we find? So we'll put in blue the ones that we can find. In red, the ones we can't find. And in purple, the ones we find, but also find additional modifications that are not. And what you can see is that we can calculate essentially most of the research that has been done on that in the past 6 years, just one reasonably sized data. There's a few residues that are not. These are phosphorylations on series.
Can I just pause you 1 second? Apologies, the sound is just deteriorating. I'm just going to reshare the slide, as I think is a problem. Be 1 moment. Okay, hopefully it should be back going. You'll have to request control again. Sorry about that.
Yep, okay, done. It wouldn't be a good online lecture if there weren't any technical issues, of course. But thanks for that. Anyway, so what you can see is we are capable of identifying all of these modifications that are well known just from 25 million spectra from across the human proteome. Now, you can do the same for something like elongation factor 1 alpha 1, where we actually look at UniProt and we know which modifications are found. So these are all the green lines with yes, and we can actually find exactly what is being predicted. What is interesting is you see the carbamidomethyl. Carbamidomethyl indicates a free amine in those residues because it's afterwards modified artifactually by carbamidomethyl, but you find it either free or with a dimethyl or with a methyl. And here you can see this lysine, for instance, is never really free. You only find it as a dimethyl or a methylated residue. And then there's 2 that are not known to be methylated, and we actually find very specifically a trimethylation on this lysine, K255, and we find a dimethyl and a trimethyl on lysine 273, but in both cases we find it free as well because it also is found with carbamidomethyl. There's only 1 lysine that we don't cover, which is the second lysine in the residue, but it's part of a very short peptide, which we're not expecting to identify or even find with mass spectrometry. Right, so IonBot actually gives us lots of data, lots of PTMs. So one of the things we've been thinking about is how do we disseminate that to the community? So we've built a system called Scope3P, which you can see at this URL, and it actually lists phosphorylations. So it's a reanalysis of public proteomics data, looking for phosphorylations and then cross-referencing them with what is known in SwissProt and mapping them onto 3D structure, as well as on a linear sequence. So very briefly, we took data from the PRIDE repository. We took 2016 raw files comprising 60.2 million spectra, ran the whole thing through IonBot. We came up with 19.2 PSMs, which is kind of 1 in 3, which is what you expect. We've got 140,000 odd phosphosites, 94,000 of which has a phospho-RS localization score of more than 0.5, and we have 14,000 odd phosphoproteins carrying these different sites. And then if you look at the system, it will tell you everything it knows about these phosphorylations. How often do we find them? Which PRIDE projects do we find them in? Are they found elsewhere? On the on the Swissprot annotations, right? So how do these two correlate? And then of course on the 3D structure, you can see the sites mapped. And then we also have biophysical predictions for every single residue, which show you things like, is it a helical region? Is it a loop region? Is it a early folding or a late folding, etc. residue? So it really helps you map modifications onto structure. Oh, by the way, if you're interested in following any of these links or finding the background information, the whole presentation will be, or is being recorded, will be made available on YouTube, and you will be able to pause that and get to these links and this background information at your leisure. Now, we're now running IonBot on a much broader scale now. We're actually trying to do it on all human spectra contained in the PRIDE database. You can see a growth panel here, a growth chart of PRIDE, which is completely through the roof. They went over 500 data sets per month during the Corona period, which is crazy. That's an enormous amount of proteomics data, but it's really nice to see. We've now processed, as a kind of a proof of concept thing, it's not yet the final thing, 25,000 raw files, which comprise 1 billion spectra. It's probably the most comprehensive human proteome ever analyzed. Ran it through IonBot, and we found 117 different PTMs on almost all proteins, and we've got almost 1 million PTM sites localized. Now, with that information, the idea is to now build a system that does more than phosphorylation. So Scope 3P is out there and it does phosphorylation, but we can actually make it do all these other kind of modifications as well. This is an actual system actually running in my group, but the problem is it's a prototype, which you can see because they misspelled phosphorylation. That would probably not make it to the final release. And then you can highlight residues based on color for different modifications. And of course, you have all the biophysics and you have the mapping with Swissprot. So we're hard at work at doing that for about the 2.8 to 3 billion spectra that we have for human data. And that's going to come out in the next months. So that is something that is that we are hard at work on right now, which is very exciting. Finally, very briefly, we can also take this one step up and start thinking about how do proteomes match with tissues. And then one way to do that is to try and predict the tissue based on the identified proteins alone, which is something that we have been doing and is very, very successful. What you can see here is the predicted tissue, and here you see the real tissue. And then you can see here, and again, this is all done on public data from PRIDE. And here you can see the very strong correlation in what the system predicts versus what the thing really is. However, it's not just perfect predictions. There are some predictions that are off, as you can see here, some of which are, let's say, 10% possibility that it is this tissue, and then the majority, the 80% vote is for this kind of tissue. So let's have a look where these things go wrong, because that is usually the most instructive. So most of the time it goes perfectly right, and the majority vote tends to be right for everything except this guy. And so we're going to have a look at where does it go wrong for the minority vote, and where does it go wrong for the majority vote. So let's start with this reasonably strong example of a minority vote going slightly wrong. Well, it turns out that the real tissue is colon, or the real tissue is rectum, or the real tissue is gut, but the prediction in each of these cases is colon. So of course, the difference between rectum, gut, and colon is essentially small enough to assume that this is real. This algorithm has figured out what gastrointestinal tract cells look like. Now, one slight odd dog out is this very weak minority opinion for a urinary bladder tissue, which is very strongly associated with urinary bladder, but which also gets some matching with colon. Now, both of these are high turnover tissues. And so it could very well be that what we're looking at is certain resemblances between the urinary bladder epithelial tissue and the tissues that you find in the gut. And the algorithm is telling us there is a link here. So this is very interesting. Even when it makes mistakes, it tends to make mistakes that make sense. And when an algorithm, especially a learning algorithm, makes mistakes that make sense, it has actually learned something valuable. Another example of this is a very weird example is a kidney and nasopharynx. So of course, nasopharynx and kidney do not seem to have much in common. Now, it should be pointed out that nasopharynx is a very, very small and specific data set. I think there's only one. So it's very easy for the algorithm to mistake this, and it does. It makes a complete mess of this. However, it matches this to kidney. And so you could say there is no obvious link, it's just a random assignment, but maybe it isn't. I haven't looked at this, I should look at this, but I haven't made the time yet. But one thing that nasopharynx is known for is GPCR receptors. And because our nasopharynx is very good at chemosensing. It's sensing, you smell stuff, you taste stuff. That's what our nasopharynx is really good at. And so it's chock-a-block full of these receptors. Now, our kidneys are chemosensors as well, because they measure what your blood looks like and then decide on whether or not they should be purging stuff from the blood as well. So it could very well be that certain of these GPCRs, which are highly abundant in nasopharynx, are also found in kidney, and chemosensing is the thing that groups these two together. But again, this is pure speculation, I could be wrong. Here's another example, the reproductive tissues. So ovary gets predicted to ovary very strongly, but testis also gets an ovary prediction, a very weak one. But again, the system is learning that reproductive systems, reproductive tissues have something in common. The link is very weak because ovary looks like ovary and testis look like testis, but nevertheless, they kind of look like each other as well. There is something here that binds them. Now, maybe a final thing is when we added a finer grained level than tissues, which is cell types. And so what you can see here is CD4 and CD8 T cells is kind of hidden behind this bar, but CD4 and CD8 don't really look that much alike. So proteomics can tell them apart, which is pretty cool in its own right. But then you can see that if there is any confusion, it's between CD8 cells and spleen. And apparently, this is something I read on Wikipedia. I haven't gone deeper into it than that. But spleen apparently is characterized as an immune-related tissue that has a strong presence of CD8 T cells. And so maybe this is something that the system is figuring out as well. And the thing I dare to dream about is something like proteomics-based digital tissue deconvolution, figuring out from the proteomes of individual cell types what the tissue looks like and how this tissue is populated by cells and how these cells contribute to the functioning of the organ, because proteomics gives you proteins and proteins give you function. Anyway, with all of that, I hope at least to have shown you that if we use machine learning to change our perspective slightly, our submarine, of which we first saw only the periscope, we start seeing a whole lot more. I'm not saying we get a clear picture yet, but we're starting to see a whole lot more. And this is just the beginning of all of this. I think we can actually get even better because we're only taking the first few steps towards this new way of doing things. So with that, all I have left is to thank all of the people in my group who actually do the real work, of course. So I showed you work from Robin on the retention time prediction. I've shown you work from Ralph and Sven de Groeve when it comes to both IronBot and MS2Pip, which Kevin is also working on. I've shown you work from Padmanabhan together with Natalia and a former developer, Demet, who've worked very hard on Scope 3P. I've shown you the tissue prediction, which is also work done by Ralph, and now with Tina, we recently joined the group, and then all of these other people who do fantastic work, but I didn't have time to talk about today. And then the very final thing I will say is, if you would like to have your picture here, we're actually looking for a postdoc in computational proteomics. So if you're interested, you can always get in touch, because we're looking for talented people to join our nice group. So thank you very much for your attention. I apologize for the slight echo halfway through, but thankfully Harry solved that very nicely. And then I think I'll hand back to the chair for your questions.
Well, thank you very much, Lennart. You cannot hear the big applause, but you get mine. And I think you took the right perspective and I think really gave us a very deep insight into what you're doing. And I also have to say, if you haven't checked Slack yet, do it, because I'm also quite impressed by one of your co-workers already answering some of the questions online with links to the tools, how I can use that. So I will only read out the questions that don't have answers, go on Slack and find the other ones. So some of the questions were brought up. One of them was a question, how about the prediction of modifications related to formalin fixation of proteins?
Yeah, that's a very interesting one, because of course, if we can get to these tissues, the whole biobanking world opens up to us. Unfortunately, we haven't actually tried these in detail, but from what I know of the chemistry, there is a high probability of crosslinks occurring. And so if crosslinks are indeed quite rampant in these tissues, we are in a lot of trouble. because crosslinks make chimeric peptides, and chimeric peptides are an absolute nightmare. Not just because of the combinatorial the combinatorial explosion of possible sequence matches, but also because the fragmentation is really bad. We've done quite a few cross-linking studies on traditional cross-linking, and what you can see is you get fragment ions from the one peptide as well as from the other peptides, and then if one of the two is rather short, it becomes extremely difficult to disambiguate which peptide is linked to which peptide. So in a way, now that I think of it, maybe for these kind of peptides, it's not so bad, because if you get at least one of the two peptides and you don't really care about the smaller cross-linked one, you could just erase that and say it's an artifact anyway. I don't care about the actual cross-link side. I care about at least knowing one of the peptides. That might help. Then again, if you get all kinds of other modifications like esters or things like that that happen, in principle, if they look like something that we have seen before, we should be able to predict those quite well. But it's a very interesting question. We haven't done it yet, but it's definitely worth having a look and see how DeepLC performs on these particular modifications. If anybody ever wants to send us some data, preferably HCD data, that would be quite interesting.
Yeah, great. There was another question, and I think it refers back to where you're comparing what this predicting spectra for different methods of fragmentation. And the question was interesting. Could you also talk about the last point where CID outperforms the native model?
Right, let's go back to that slide. If I quickly press back very many times. Because that's the triple TOF model, I believe, that was there. I think what you see there is just a training artifact, in that we did not have as much TOF data as we had CID data. And it appears that CID data is reasonably similar in fragmentation outcome to TOF data. So as a result, the larger data set, which is the TOF data set, actually has a slightly better prediction accuracy than you would expect. But in the end, what you see here is if it's this one here, the triple TOF does perform best on its own data. And the HCD model, actually looks a lot like the triple TOF model here. Again, probably because the TOF model should be able to do better, but we just didn't have enough data when we trained this. And having more triple TOF data would move the brown point up, whereas the orange dot probably would stay put. That's what I think would happen here. But I didn't discuss it in detail because, again, triple TOF, we didn't have that much data. And you can see that the actual prediction accuracy is below par compared to all the other data sets. Yeah.
So related question also on these topics, would you still expect the issues you described with TMT if one was acquiring TMT using SPS MS3 and the MS2 is giving you the peptide sequence info, or the MS3 the quantification issues?
Well, yes,
I don't know, clear that there's more explanation in there, but
yeah, so the MS3 step essentially is a really great step to try and get a clear noise-free signal from the TMT label. So we did quite a bit of work on that in the past, and I know about these overlaps and the fact that you can get noise added to the real quantification, especially on the lower amounts. And so the MS3 really helps you distill the signal from the iTrack label. Having said that, at the MS2 stage, still the fragmentation of that peptide is going to be affected by the presence of the label. So I again would hope that the TMT model will predict much more accurately and could offset the identification bias you might have, whereas then the MS3 level would give you a good quantification. But I don't really expect the fragmentation to be that dramatically different. Having said that, I haven't checked it in detail. But my expectation is that it would not matter at the MS2 level.
So now, because it's a related question for myself, so I've been wondering, because now you can probably tell which way exactly the TMT label affects the fragmentation, right? Because in the end, it's kind of chemical, physical properties. And I don't know whether you can extract from your data what exactly is happening there. And also with the TMT Pro, whether the same thing would happen or whether that would change the fragmentation in a different way.
Yeah, so in principle, it is possible with the algorithms that we use to predict MS2 patterns. It would be possible to figure out at least which characteristics are important. However, we have not done that on TMT. We have not specifically looked at which things are more important. I can tell you in generic terms that what matters is for shorter fragments, the ionizability is very important, as you would expect. So the capacity to hold the additional charge safely. Because, of course, smaller ions that have a charge and can't really hang on to it thermodynamically very well, they tend to break or lose the charge. And when they lose the charge or break, they disappear from analysis. As soon as peptides get a little bit longer, what we saw was helicity gets more important. Very interestingly, you see that when you have 4 residues or more in the fragment ion, which is exactly 1 turn of an alpha helix. So that's exactly where you would expect helicity to become important. Why? Because it probably stabilizes the peptide while it's flying. So now it's difficult to break it again. And so that probably helps there. So we can get this kind of information out of this. And in principle, we should be able to do it for TMT. And then if we would get that information, I could give you a sensible answer on the TMT Pro. However, we haven't done this, so I can't give you that information, but it's definitely worth looking into. Because again, this is not a deep learning network. So we actually can interrogate the model and figure out what kind of parameters are important to the fragmentation.
So we'll give you one last quick question, and perhaps it would be great if you can continue asking the other ones on Slack. And again, your coworker has done most of them already. It's just quick. How does the DIA version of MS2 PIP compare to commercially available software tools, for example, Spectronaut?
Yeah, so we haven't done that direct comparison because we don't have the commercial tool in-house. However, I can tell you that the Kuster group, together with people in the US, with Brian Searle, have done a very, very similar thing. And when we actually compared the results that they got versus the results that we got, they're extremely similar, despite the fact that the algorithms being used are very different. And so this is what I said earlier on. I think we've reached a level of sophistication with MS2 predictions that are probably on a diminishing returns curve. So we can make the algorithms 10% better, which is going to be very difficult and take a lot of time, but it's only going to make, say, 0.5 or 0.3% difference in the final results. So I would imagine that any algorithm that is being built today that is somewhat competitive with what is out there will have very similar results. What is quite crucial, however, is the retention time. So the accuracy of the retention time prediction is a key feature. And so there you have 2 factors. You have the accuracy of the algorithm itself and the calibration of that algorithm to specific LC setup. So this is in generic terms. Again, we haven't tested it against Spectronaut specifically.
Yeah, great, thank you. That was really, really very helpful and useful. Thank you for your presentation and for answering the questions. And now for everyone who needs a certificate, the slide will come back where you can scan the code and get those for yourself. So with that, I would like also to hand the presentation back to Jay.
Great, thanks Christian and thanks Lennart. Thanks both really a lot for your time today. An excellent talk, Lennart. Yeah, as Christian said, hopefully shortly on the screen you'll be able to see the QR codes for scanning for your attendance certificates. As Lennart mentioned, everything's recorded and will be on YouTube as soon as we can get it up there, so you can go back over his slides and pause the talk. And hopefully Lennart and his colleagues will take a look at the Slack channel questions, which we didn't manage to find time to answer for, and we'll answer them in there. So keep your eyes on the Slack channel. Yeah, great talk, Lennart.